Hey.
Good morning, good afternoon, folks. It's Mark Gardner here. Welcome to this Global Health Compound Design meeting. We will get properly started in a couple of minutes, just waiting for a couple more people to get connected. At the moment, I'm hoping that you should see a poll question, which is asking about your use of predictive models. I would like to have asked in more granularity, but the technology only allows me 5 options, so you've had to choose one of those 5. If you haven't voted yet, it would be great if you could. Just interested to see your answers. And so the agenda today is going to be with 2 speakers. Professor Jonathan Goodman is going to kick us off. He's going to talk about solubility prediction, outcomes and insights from the Solubility Challenge competition. He's from the University of Cambridge here in the UK. And then Matt Segall from Optibrium, also in the UK, is going to talk about how confident can we be in ADMET predictions, which I think, some of you may have responded to our recent survey. Many thanks for doing so. And it was clear from the survey that people are interested to learn more about both where they can get a hold of models and also about applying models. So this presentation today will be very timely. Just a reminder of a couple of the housekeeping items. If you want to speak, please click on the hand icon in the attendees panel. If you want to communicate with us, the question pane is probably the best way to do it. And if you're asking scientific questions, I know Jonathan and Matt will do their best to answer them, but if you have other points as well, by all means, raise them. So I'm going to suggest we now kick off with Jonathan Goodman. So thanks very much, Jonathan, for agreeing to present. If I can, wait a minute, I think I need to close this poll. So let's see, are there any last people wanting to vote in our poll on using predictive models? Last chance before I close it and everybody gets a short view of it. So hopefully you can see the results. Oh, no, I have to share it as well as close it. Hopefully you can now see the results of the poll. Okay, so a third of you using quite a few models, a quarter of you not using any, and some ranges in between. So now if I hide that poll and change presenter to Jonathan, all should be well. So Jonathan, thank you very much.
Thank you, Mark. I'm just seeing if I can produce some slides. Can you see my first slide?
We can.
Yes. So thank you for the invitation to talk to you about some of our adventures in solubility prediction and measurement. I can't currently see the box which says questions, so if there is a question, perhaps, Mark, you could tell me. So do please ask questions as I go on. Because we became extremely interested in solubility about 10 years ago and thought it couldn't really be all that difficult. So we very rapidly realized that really it was an incredibly challenging problem. And so I can tell you about a few of the things that we've done over that time. If I can work out how to change the slide. Oh, yes, here we are. So the first thing, of course, is you can't get a good calculation unless you have good measurements. And so we thought we could just read the literature, though it turns out the literature isn't terribly reliable on solubility. So this is an outline of the things I'm going to go through, how we managed to make measurements, combining different information sources, then making our own measurements, and then talking about how we try to predict them and how we and what we think the future might bring. So to begin, how can we measure solubility in the first place? Well, the traditional way is just shaking flasks, which works rather well, but it's quite difficult to tell if you've reached equilibrium. It's very difficult to control it very precisely, and it's quite difficult to measure the concentrations. there are much faster, high throughput ways like measuring turbidity. So you have a solution of something which is dissolved, add water, and see what happens. And this is great because it's quick and you can automate it, but it's not clear precisely what it is that you're measuring. Probably a better way is varying temperature and seeing how solubility changes with temperature. But this is terribly slow, and again quite difficult to work out exactly what it is that's happening. And the way that we decided we liked most was by varying pH, a potentiometric method, which I'll say a bit more about in a moment. But we realized that to begin with, if you say measuring solubility, you might mean a huge range of different things. So people report something they regard as solubility, but it might be kinetic or thermodynamic or equilibrium or ionic or intrinsic or aqueous or standard or natural, or all sorts of different things. And all of these can be measured for the same compound and give really dramatically different answers, all of which are right. But if you don't know, or all of which could be right, but if you don't know which it is that's being measured, then you're rather stuck. And often papers don't report in enough detail exactly what the measurement was. Quite small changes in temperature can lead to big changes in solubility. Changes in dissolved salts, which are nothing to do with the thing you're interested in, can also change things in quite a big way. And pH, of course, has a really very dramatic effect on solubility. So we decided that we would focus on the intrinsic solubility, which is the solubility of the neutral form of ionizable compounds. Because many compounds, and most drugs, in fact, will ionize if you put them in water. And the ionized form is usually, though not always, really rather soluble. It's the unionized form which will lead to precipitation. So if we can work out the intrinsic solubility, the solubility of the unionized form, then we should, in principle, get a pretty good measure of the solubility at different pHs, as we can work out the amount of the substance which should be protonated, and so how much ionized form there is there. So intrinsic solubility sounded like a good thing to measure. And if we can measure the intrinsic solubility and do it reliably, then we should be able to produce a wonderful model of how it all worked. At least this was our initial hope. But of course, this was something of a vain hope, because we'd already tried similar things in other areas. For example, looking at NMR spectra, we already have, there are extremely good models of NMR spectra. You can put a compound into Gaussian, do a calculation, and get out a spectrum which looks very much like the experiment. So on this slide, if you look at the top left-hand diagram, you can see the calculated NMR peaks plotted against the experimental peaks, and most people would agree this was a rather wonderful straight line with an R squared of 0.995, and I think I could convince most people this was a really very, very good correlation, and maybe that this demonstrated that the experimental and the calculated structures corresponded to each other. However, it turns out this isn't actually the case, that the calculation, the calculated spectra was calculated for a molecule which wasn't the molecule for which a measurement was made. So on the bottom left is the molecule which the calculation was, and on the bottom right is the molecule for which the experiment was made. And if you compare the calculation for the correct molecule against the experiment for the correct molecule, you get an even better correlation. So an R squared of 0.998856 instead of only 0.995. So if you can see both of these, you might believe that the right-hand one is better than the left-hand one. But essentially, they're both very, very good, and you can't necessarily convince yourself one is better than the other. So in order to simulate things in a useful way in NMR spectra, you have to be very, very precise. In order to simulate things in a useful way or predict things in a useful way in solubility, then we can probably get away with a lower precision, but we're still going to be quite worried about what it is that we're actually measuring and how we can interpret our results. And experimental data, can we ever believe anything in the literature? Well, we can probably believe quite a lot in the literature, but not as much as we'd like to. A little while ago, we wrote a program together with Peter Murray-Rust and colleagues at the Royal Society of Chemistry, which read chemistry papers and tried to put in checks to do fairly simple cross-validation to see if the experimental data in organic chemistry papers really can possibly be consistent. And depending how you interpret the results, you could say this showed that most chemical papers were pretty good, or you could interpret it as saying probably many and perhaps a majority of published chemical structures, published chemical reports have a few mistakes in, which might worry you quite a lot. So experimental data, even for things which are quite straightforward to measure, like, for example, NMR spectra, are not 100% reliable. And experimental data for things which are very difficult to measure, like solubility, are going to be dramatically less reliable. So we were worrying more and more about the quality of the data. Maybe we should have been worrying less, because if no one knows if our answer's right, then we can just have to concentrate on selling our model rather than actually getting it correct. But yeah, but ideally, we would get the right answers for the right reasons. And we would store all our data in a nice ordered XML data set, because this is the right way to do things. And then we'd like to calculate things too. And in principle, it's very easy to measure, to calculate solubility. All you have to do is get a solid, calculate the free energy of melting, calculate the free energy of boiling, then calculate the free energy of hydration, and subtract the free energy of hydration from the free energy of melting and the free energy of boiling, and you get a measure for the free energy the free energy you need to calculate the solubility. So this loop all looks very good, but unfortunately, the free energy of hydration and the sum of the free energy of boiling and melting are both very large numbers. So you're calculating two very large quantities, which you can do reasonably accurately. You then subtract one from the other, and so the error in the difference is huge and probably larger than the difference that you're actually, than the precision of your calculation. So whilst this approach does work, and I've put on the slide a paper where John Mitchell was leading this study of doing this sort of thing, it works reasonably well, but it doesn't work anything like as accurately as we'd want it to. So this is a sort of ab initio calculation, which is promising but not perfect. We could also begin from molecular descriptors and try and work out from molecular descriptors what solubility is likely to do. And that also works reasonably well, but not as precisely as we'd like it to work. So all of these assessments on how accurate they are, of course, depends on whether we believe our experiments. And we decided to measure intrinsic solubility using a machine we bought from Sirius, which is an analytical chemistry company based near Gatwick. And they have a wonderful machine they call the GLPKA. And this is a terribly clever machine because it can control the pH of a solution rather precisely. And that's good because if you have an ionizable compound, by very slightly changing the pH, you can slightly change the amount of unionized compound. And so a tiny tweak in pH will change you from being a subsaturated solution to a supersaturated solution. So if your experimental system is a mixture of the ionized form and the unionized form, and you slowly adjust the pH, you can count how many hydrogens are bound on average, and so see how and so work out from a measurement of the pH and from knowledge of everything you put in, exactly how much unionized form there is. And you can see by the changes in the pH whether things are precipitating or whether they're dissolving. So here I have a slide for a measurement of the pH of our solution against time for diclofenac, which many of you have probably eaten. So at the beginning of the experiment, we begin with a solution of diclofenac. We drop the pH until it begins to precipitate, and then we raise the pH again until we're getting to the point where it's just precipitating at one pH and just dissolving at another pH. And these two pHs are really very close to each other. So the area which has been circled in green, which looks more or less like a straight line, is changing between pH 7.10 and pH 7.13. And this tiny tweak in pH changes it from a subsaturated to a supersaturated solution. So we can bracket the equilibrium solubility, the equilibrium intrinsic solubility rather precisely. And this rather sophisticated approach from the Sirius company means that we can get a reproducible and thermodynamically justifiable measurement of pH. So we really enjoyed doing these measurements because they were, we felt, much more reliable than had been done before. and because we could use the same results from a wide range of different samples, reproduce the results if we tried the experiments at different times, and define in a sufficiently precise way exactly what it was that we were doing. And this mattered because if you look at compounds even like caffeine, which isn't a particularly unusual compound, then its solubility as measured in 1926 was 2 grams, and its solubility as measured at the end of the 20th century was almost 1 kilogram per liter. And it's very difficult to see how these 2 different measurements can possibly be of the same compound, and yet they were both published in reputable journals at the same temperature, though presumably not with the same attention to whether this was kinetic or thermodynamic, not with the same attention to exactly what the precipitate was, maybe slight differences in pH or slight differences in dissolved salts. So there are all sorts of reasons why this dramatic change in solubility may really reflect experiments which were done carefully and with good intentions. But if your measurements vary by a factor of almost 2 orders of magnitude, or almost 3 orders of magnitude, then we do have a bit of a worry for the calculation. We looked carefully at diclofenac, the compound I'd shown you before. If you search diclofenac on the axis, then there's a button to give you the solubility values. And the solubility of diclofenac in the axis appears as a big table, which varies over 3 orders of magnitude for the solubility. Of course, if you read it a bit more carefully, you discover that they're measuring the solubility in different ways. So some are intrinsic, some are at low pH, some are at high pH. So many of these experiments are probably correct but hard to compare. But because we were measuring the intrinsic solubility, we could do this by taking as our starting material a sodium salt or an acid form of diclofenac. We could measure the solubility really very precisely, and we came up with an answer which was similar but slightly different to other to previously published intrinsic solubility measurements of diclofenac. And as it was precipitating, we could characterize our solid forms either as sodium salts or as pure acids. So this gave us a really good measure of one compound, though clearly any compound which, any measurement of just one compound is not going to be totally interesting to simulate. And we discovered that changes in solubility were really quite dramatic and unpredictable. So on the next slide, a sequence of 2 series going from, if you look at the top left, a secondary amine to a methyl secondary amine to an ethyl secondary amine. None of this looks really terribly interesting, you might think, but actually the solubilities just oscillate wildly, and you might expect that methylation and ethylation would lead to some sort of sequence or trend, but it really doesn't. And the trend for the top row and the bottom row, or the random pattern is different for both of these series. So clearly, simply making small conservative changes to a tiny part of the molecule doesn't lead to a small and predictable change in solubility or the intrinsic solubility. But even if this is if we're just worried about trends in solubility, we also have to worry about polymorphism, that a single substance can have multiple crystal forms. And Macron's law, which I rather like, is the number of polymorphs a compound has is proportional to the amount of time that's been spent on it. So clearly, the more people study compounds, the more polymorphs there are, and almost certainly there are many, many more polymorphs than people have ever identified. But it turns out that by using our tensiometric method of solubility measurement, we could also define which polymorph we got in some different cases. You can get polymorphs by lots of different methods which I list on this slide, but if you use a potentiometric method, then we found we were beginning to decide on one solubility for a compound, and we've just about converged there, and suddenly it would leap to a completely different one. So sulindac, which is another rather common compound, the solubility changed by a factor of 7 as the experiment progressed. And because this was a reproducible effect, we could, so if we stopped the experiment quickly, we got one polymorph. If we waited longer, we could get the other polymorph. And this was a good case because we could define what was going on really rather precisely. But we had many other cases where we had a similar sort of effect, where we were getting localization on one value, and then it was suddenly jumping to another, and then suddenly jumping to another. So our most exciting one seemed to have 7 or 8 different solubility measurements. Unfortunately, some of them were so transient we weren't able to isolate the intermediates. But what do we mean by solubility when there are half a dozen or more different things which you might be able to isolate? Is really a very difficult question. So is this all hopeless? Well, not quite hopeless. We had gathered a large amount of data on different compounds which we thought the world would be interested in. So we then tried to publish it, which seemed like the natural thing to do. And it turned out publishers were not very keen in publishing a paper with fundamental useful data in, because it doesn't have the story which is expected in some for most journal articles. And this seemed to me to be a real problem with publication systems, if you can't publish data as a paper and make it available to people and hope people will cite it. So our way around that was to persuade the American Chemical Society, who very helpfully agreed that we could run a solubility competition instead of just publishing our data. So to make our really exciting database even more interesting, we published a database of 100 reliable measurements. Well, reliable in the sense we'd done them ourselves and we sort of believed in them. And then we gave the structures of 32 more molecules for which we'd also made the measurements but hadn't actually hadn't actually published the results. So we thought this might be a paper people would read and maybe cite, but also they might try the experiment, so tried to enter our challenge. So we published this in the Journal of Chemical Information and Modeling. It was advertised, and people sent their answers into the editor of the journal. And shortly afterwards, we were able to publish a paper which gave us the findings of this challenge. So we think our database was pretty good. So here the blue blobs are the scatter plots of solubility against molecular weight for the 100 compounds in the training set. And the red blobs are the same scatter plot for the 32 in the test set. So on this fairly simple simple impression of diversity, it does seem to be fairly well distinguished. Did it actually work? We made all this data available, and it's still available if you'd like to copy things. If you'd like to try this again now, maybe the answers are more easily got now because, of course, we've published 32 solubilities as well as 100. But this is still available, and do please try it. Did it work? Well, the answer could be either yes or no, depending how we look at it. So we don't know who entered this competition. We asked them all to give themselves a 5-letter code name, which you can see along the bottom of this table. If we say, in order to get the right solubility, we'll only count it as correct if it's right within 10%, then you can see people really didn't do terribly well. So the best people, the best models, we're getting about 20% of our solubilities correct, provided we said provided we were saying you have to be right within 10%. So maybe this criterion is just a bit too tough. And actually, if you look on the left-hand edge of the table, we've no idea who sent in these different acronyms, and anyone was allowed to choose their own acronym. So the team which got 0% correct, just because its acronym is ASTRA, doesn't mean it's actually connected with any specific company. But this, so this looks quite bad. So if we look at this table, I think we'd say it's impossible to predict solubility. But if we try and spin the results a different way, and say, actually, you've got the solubility correct if it's correct within half a log unit either way. So this is within an order of magnitude overall. And that perhaps is a more realistic test. It would be nice to get precise solubilities, but if it's right within an order of magnitude, that would be useful for medicinal chemistry. It will tell you, ask the question, is this soluble? Answer the question, is this soluble enough or not in many cases? So if we have this much more lax impression of what's good, then quite a lot of methods were really getting things right within half a log unit or so, and doing that most of the time. So clearly it is possible to predict solubility. We don't know what all these algorithms were, and to do it reasonably well. The 32 compounds we asked people to test were these 32, and they fall into groups. The groups in the blue box, which nearly everyone got right, the groups which are in black and white, which a lot of people got right, and the groups in the yellow box, which most people got wrong. And it turned out that the groups in the yellow box were all measured to have really rather low solubilities. In fact, on the next slide, this graph, if you look at the bottom right here, the blue bar shows the solubility, the percentage of people who got it right, and the red bar shows the solubility of the compounds. And as you can see, the ones which people tended not to get right are the lower solubilities on the left-hand end of this curve. So terfenadine, fluocinolone, and propenicid are some of the least soluble compounds in our study, and people tended to get those wrong. And towards the right-hand side, where the solubilities are higher, then people were more likely to get a right answer. In fact, you might think that if you just guess that the solubility of everything, or log solubility of everything, is 2, then actually you do get a pretty good overall result, which is also slightly perturbing. You might notice that one result stands out here, indomethacin, which has which almost no one got right, even though it has quite a high solubility. And at first we didn't really worry about this. We just thought how hard it is to do this. But later on we did begin to worry about this a bit more. And we realized that indomethacin, the structure at the top right here, looks like a perfectly sensible, stable amide. But actually this isn't a normal amide, because it's not possible for the amide structure to be flat. It must be twisted through 90 degrees, which means it's a really very reactive amide. And this means that if you put it in a moderate pH, it falls apart. And so we had made a mistake in our experiment, even though we'd done it terribly carefully, because even though we were putting indomethacin in, what we were actually measuring was the solubility of the hydrolyzed form, which had turned into a carboxylic acid and an indole. And we were measuring 2 different solubilities in one experiment, so it was all terribly clever once we realized what we were doing, which unfortunately we didn't at the time we published the solubility challenge. So and many people were getting the true solubility of indomethacin about right. So the attempts at predicting solubility are slightly better than they looked. However, the same general trend remains with just these small changes in with a single result being corrected. So overall, solubility is incredibly complex. We are able to predict it, we're able to have some idea of what's going on to some level of precision, but we'd like to have much better methods of prediction and much better methods of measuring the solubility, because the uncertainty in the measurement, which perhaps is really limiting the future development of models. So with that, here are a few key references, and I'd be happy to answer any questions.
Fantastic. Thank you very much, Jonathan. I'm not sure I shall ever try to predict the solubility again, although I guess some people are right some of the time, so that's good. Now, there was a question from Anthony. Anthony, do you want to ask it? I've just unmuted you.
I was just wondering 2 things. Why is carbonic not soluble in water? It's sulfonamide and it's carboxylic acid. It should be able to dissolve pretty easily. Also, are most drugs given as a hydrochloride salt that assumed to make them more soluble and to ionize them?
For propenicid, yes, it does look as if it ought to be quite soluble, but its solubility isn't all that high, and I've learned not to be surprised at anything. So I'm now not surprised at its solubility. Of course, and I'm certainly not surprised enough to think that the measurement is wrong. I think that measurement is correct. It just, qualitatively, the reason quite likely that it has this carboxylic acid and the sulfonamide, it may be in the crystal structure. They just stick together really well. So it's not so much its free energy of solvation is surprisingly high, low. It's more that the crystal structure is surprisingly stable. And given the choice of being in solution or in a really stable crystal, it just likes to be in the crystal. So qualitatively, but I'm speculating, that's not so surprising.
It's not so much energy as it is the actual crystal structure that can cause problems.
Yeah. And then the question of delivering them as hydrochloric acid salts, well, that's often the case. But if it's, but that changes the pH. So if you have a solution in a vial, you can add hydrochloric acid, lower the pH, and they will become more soluble. As soon as you inject it into a patient, then the pH changes from what it is in the vial to the pH of blood or whatever, wherever you're injecting it. And so there are cases where the sudden change of pH means that the drug precipitates inside the patient, and that's not good at all. So making it a hydrochloric acid salt may help before you inject it. You really have to worry about what's happening when it's in essentially a buffered solution inside the body. So it helps, but it doesn't solve the problem, and you still have to be very cautious.
Fabulous, thank you. Just a reminder, if people do have questions, by all means raise your hand or enter them into the question box. I did have one other question, Jonathan. With all the different types of solubility that you mentioned, you're measuring intrinsic solubility, if I'm understanding that correctly.
That's right, yes.
And so the intrinsic solubility, the way that you're varying the pH around the point at which you're getting supersaturation, presumably is this fairly similar to, ultimately, I guess one would assume that this is relatively similar to a thermodynamic solubility at that sort of, I don't know.
Yes, it is a thermodynamic solubility. Well, given, one has to be careful about these terms because we can, as I said, begin to converge on a polymorph. So it's not truly thermodynamic if there's a lower energy polymorph. So you may be in a kinetic trap, but if you're in a situation where you can flip very quickly from going to dissolving to precipitating with a tiny, tiny tweak in the conditions, then you're bracketing something which very often will correspond to a thermodynamic solubility. We can measure kinetic solubilities too, and the uncertainty in the kinetic solubility is about an order of magnitude greater than the solubility we measure by this cycling approach.
Interesting. And Tim, I think you have a question?
Yes, hi there. This is a very nice presentation. My thought was that having a test set of about 100 compounds to work with is very nice if that data is of high quality, but still 100 compounds is not much to work with. Are there any prospects or plans of to extend this into a much larger data set that can be used and publicly accessed?
We then, unfortunately, our funding ran out, and the 100 compounds took quite a long time to measure. We'd be very keen to create a much larger database, and if you have any suggestions for finding ways of sponsoring this, then I'd be very keen to talk about them. But the research councils have not been terribly keen in the UK, so I think it's partly because they say it's something which is directly useful for the pharmaceutical industry, so they should be funding it.
Right. Thank you very much. I think we should move on. But thank you very much indeed, Jonathan. And I'm sure again, if you have other questions, folks, by all means put them in the question box. And you know, if we don't get time to answer them today, I'm sure Jonathan will be happy to try to address them by email or something. So I will now make Matt presenter.
Excellent.
Okay.
Here we go. Hopefully you can see my screen, and now hopefully you can see my slides.
Yep, that's perfect.
Can I just check you can hear me okay as well, Mark?
Yes, that's great. Thanks, Matt.
Wonderful. Well, thanks for the invitation to speak today, and thanks to everyone for joining us. So I guess I'm going to pick up after Jonathan's very nice presentation about solubility to speak about ADME predictions in general. And the title of my talk is really, how confident can we be in ADME predictions? And so what I'm going to do is to just give a brief introduction to predictive ADME, and particularly some of the methods, and then really focus on some of these key questions that we have when we're working with predictive models. How are they validated? How do we think about uncertainties, or the flip side of that is confidence in predictions? and the domain of applicability, or where those models actually can be applied with confidence and where, conversely, we shouldn't trust them. And then at the end, I'll just say a short few words about, given these limitations, how we can still use ADME models to guide optimization before drawing a few conclusions. So first of all, an introduction to predictive ADME. Okay, well, I think with the audience here, I don't really need to spend much time on why ADME or ADMET is important, but I guess just important to remember that, as we mostly know now, a potent molecule is not necessarily an effective drug, and in a gross approximation, it's these absorption, distribution, metabolism, excretion, and toxicity properties that really make the difference, because a successful drug must travel from the site of administration, get to the site of action, remain there in sufficient concentration for long enough to have the pharmacological response, and then frankly get out before causing any toxic side effects, or before causing significant toxic side effects. And so clearly in drug discovery, making sure that we get the right balance of these ADME properties is very important. And of course, one of the focuses over the past sort of 15 or 20 years has been how we can use methods to predict these ADME properties as early as possible in the process, save time measuring them, or at least when we're working with virtual compounds, getting a feeling for what the ADME properties of a compound are likely to be. And on this slide, I've summarized a few of the sort of common methods that are now applied to this challenge. The first of which are quantitative structure-activity relationship models, or QSAR models. And essentially, in a manner similar to that which Jonathan described, essentially these are based on taking a set of molecular features or descriptors and fitting a mathematical model to a set of experimental data. And a whole range of different machine learning or statistical methods are applied to perform this fitting. When we can actually sort of begin to understand the sort of chemical mechanism behind these processes or properties, then quantum mechanical simulations can be very helpful, particularly for chemical reaction-based mechanisms such as metabolism, and an area that we work in is using these sorts of simulations to look at metabolism by cytochrome P450s. They can also be very useful for some of the physicochemical properties and calculating some of the free energies in very small numbers of cases, because these tend to be quite expensive calculations. An alternative approach that's commonly used is an expert system, where you try to encode the rules that experts sort of understand and know about properties by interpretation of experimental data, and apply these rules to compounds to predict the likely outcomes. And this is particularly used for properties like metabolism and also toxicity. And then when we're interested in the in vivo pharmacokinetics of compounds, then often these physiologically based models of pharmacokinetics are used. And typically that is used to combine individual properties, absorption, solubility, metabolism, etc., and combine those in a mechanistic model to simulate the in vivo pharmacokinetics. Now, those individual properties are often measured as inputs to these PBPK models, but in some cases they can be predicted. So there's a whole host of different methods that can be applied. For the purposes of today's talk, I'm really going to be focusing on the first of these, the QSAR method for predicting ADME properties. And so in a little bit more detail, as I say, what we're trying to do here is to take a set of descriptors that capture some features of the molecules of interest and fit a mathematical function, shown here as f, between those descriptors and the actual observed values of this property. And of course there are a number of components that must go into this sort of modeling, and again, as we've heard a beautiful example of, data is where it all starts, and quality data is absolutely essential. If you start to combine data from different endpoints for different protocols or different experimental conditions, then often what you end up doing is fitting not the relationship between compound structure and these activities, but actually between the different experimental measurements. And again, we've heard a beautiful example of the fact that public data needs to be treated with extreme care, and often is not really good enough to build quality predictive models. The second element that goes in here are the descriptors. So how do we actually characterize the molecules in order to make predictions? And there are a whole host of different descriptors that can be used. We can look at sort of whole molecule properties, so log P, which is itself a model actually, of the experimental lipophilicity, molecular weight, polar surface area, and many different other ways we can sort of characterize that molecule as a whole. But often we also break that down into structural descriptors, whether that be by smarts or substructures that are of interest, fingerprints, and again, many, many other ways, both two-dimensional and three-dimensional descriptors that can be used to characterize the structures of these compounds. And once we've chosen a set of descriptors that we think capture these sorts of variations, then what we need to do is actually to fit this mathematical function. And again, a whole host of different methods are applied for this, from simple sort of statistical fitting like multiple linear regression or partial least squares, to the sort of latest in machine learning methods, whether it be neural networks, Gaussian processes, support vector machines, random forest, etc., etc. And you'll find whole hosts of papers describing why their method is better than another and so on, but in practice, you know, there are many different methods that can actually build reasonably good predictive models. But ultimately, there are some key issues and questions we need to address. The first of these is the confidence that we can have in the predictions. When can we trust a model to actually make predictions? Or can we trust it at all, frankly? And if we can, then how much should we believe or trust that model? And given these limitations, how and when can we actually use these model predictions effectively to actually make a decision? And ultimately what we'd like to do is not just get a number from these models, but actually begin to interpret them to provide guidance on, you know, key structural influences to help us to think about how we can design compounds to overcome issues with ADME and physicochemical properties. So let's think about how we can actually begin to address some of these questions and think about the reliability of models and how we can begin to interpret them. So first of all, can we trust a model? And this really comes down to the validation of a model. And so this is a sort of a quick schematic of sort of a common sort of model building workflow, where of course it all starts with that high quality data. And typically we might be looking at splitting that data into independent sets, two or often in as shown 3 different sets. I call them the training, the validation or internal validation, and an external test set. And this training set is actually used to build models, to fit this mathematical function of the descriptors to the observed properties of the compounds in that training set. Now, I could fit this in many different ways, and in practice, one could fit it with arbitrary accuracy. You can make a model that fits each point perfectly, but that model will not necessarily generalize, or often will not generalize very, very well. And if we're using different modeling methods, then the next stage in the process is really to think about what modeling method is actually giving us the best result for this particular chemistry and this particular property, because there's not one modeling method that's always best. And so here I've listed a few from the previous slide, like partial least squares, radial basis functions, Gaussian processes, random forest, and so on. And so what we often want to do is compare between different models that we've built and find out which one is apparently the best. So we can evaluate these models in terms of their predictive performance on that internal validation set. But we can build lots of models, we can compare them, we can find the one we think is best, but actually that validation process is itself to some extent training to this validation set. If you build enough models, by statistical chance you're probably going to find one that fits reasonably well. And to be absolutely rigorous, we would always suggest using an external independent test set essentially a blind test, as we see again in Jonathan's nice example, to evaluate the model we think is best and confirm that hypothesis. And what we should hopefully see is that the performance on this external independent test set is similar to the performance that we found on that validation set. That would give us confidence that that result generalizes well. And so external validation is very important. I would caution people against looking at models that have only been sort of cross-validated or internally validated. So if we have a validation against this test set, how do we actually measure the performance or assess the predictive ability of a model? And the way that we tend to use is essentially by looking at the so-called coefficient of determination, or often represented as the R squared. And this is a measure of the fit of the predictions Y to the identity line for X. The perfect model would give exactly the measured value for every prediction. Of course, that's unrealistic, but that's the ideal we're aiming for. It's important to be cautious because this coefficient of determination, rather confusingly described as R squared or represented as R squared, is often confused with the squared correlation coefficient, which I'm showing here is the R squared cor, which is actually a measure to the best fit line. So here's a quick example where you can see for this set of data that the best fit line is shown here in orange, and the fit to that is pretty good, R squared of 0.86. But actually, because that best fit line differs quite significantly from the ideal identity line shown in blue, in fact, this coefficient of determination is quite a bit lower. So we feel this is a more rigorous test of the performance of a model. And so again, be careful when you're looking at the performance of a model, which of these R squared is actually being reported. And of course, the other measure is the so-called root mean square error, which is essentially a measure of how accurate one can expect a prediction to be by that model. Of course, we don't always have the luxury of predicting a numerical value, and often we build models that are classification, high, low, or good, bad, or high, medium, low, whatever your classes may be. And so one needs to be a little bit careful about the validation of classification models as well. This is an example here where I've got a very biased data set. So the vast majority of my data set is actually observed to be high, very few are low. So the accuracy of this model is actually 92%, even though it doesn't predict low very well at all, because simply by predicting everything to be high, I can be accurate in the vast majority of cases. So one needs to be careful because very often in sort of drug discovery field, we have these biased data sets where we have a predominance of one class over another. And if we just look at the accuracy, we could be misleading ourselves. And in this case, again, there are different approaches. There's the Matthews coefficient and so on. We tend to use the kappa statistic. But essentially what we'd like to use is a measure that is corrected for this bias by essentially, in this case, correcting for the expected performance of no model. So just predicting or randomly selecting based on the distribution in the data set. And also very useful to look at the sensitivity and specificity of these models as well in order to assess how useful or accurate they will be in a particular context. So we can validate these models in a rigorous way and assess how good they are. But the next question that we mentioned is, well, how can we actually assess where a model can be applied with confidence? And this is the so-called domain of applicability. I mean, I sort of illustrated it here with a little schematic where here I've just got two descriptors because I can draw two-dimensional pictures quite nicely, whereas high dimensions are quite difficult. But essentially what I have here are the set of gray points representing the training set for a model. And I'm showing here just schematically as an ellipse, this area of this descriptor space that is well represented by that training set. That's sort of the domain of applicability of that model. And so if you have a new compound, say like this blue star here that lies deep within that domain of applicability, we can make a prediction for that compound with confidence, and we can assess the uncertainty, the expected uncertainty in that prediction in terms, typically from the validation of that model within the same space. On the other hand, if we have something like this green star, it lies just outside of that domain of applicability, the model is beginning to extrapolate beyond what it's been trained to recognize, and so the confidence in that prediction will become lower. Essentially, the error bars will become higher. And in the extreme, we might have something like this orange star that lies way outside of that domain of applicability. And in that case, of course, the model can generate a number, but frankly, we have no clue how accurate that number will be because we've got no data, no evidence to support it. So typically we would suggest that has a very high uncertainty. So you can come up with these sort of domains of applicability. There are many, many different ways of doing this as well, and lots of discussions about what the best method may be. There is a more subtle way to look at this challenge as well, and that is with some of the sort of more advanced methods. I'm showing here an example called Gaussian processes, where actually instead of just giving an in or out or in or close out of a domain of applicability, we can actually assess the uncertainty in each individual prediction. So this Gaussian processes method uses sort of a Bayesian mathematical approach, and what it does is it actually fits a distribution of possible models that explain the data. And so what you actually end up with is not a single prediction, but a probability distribution for the prediction for each individual compound, where you can make a prediction as the mean of this distribution and actually look at the standard deviation of that distribution as an estimate of the uncertainty in each individual prediction. So it's not coarse-grained, but each compound will get a different level of uncertainty depending on how well supported that prediction is by the compounds in the training set. And how does that perform? And here's an example. This isn't an ADME property, but it's a Gaussian process model of actually affinity of to an adrenergic receptor. And I'm just showing a quick summary here where you can see that for the compounds that we estimate to have a low standard deviation in that predicted value, and the vast majority of them are observed to have a low error, and we get roughly the proportion that we would expect to see, 1, 2 standard deviations and so on, whereas those predicted to have, or those with a very poor prediction tend to have a high estimated standard deviation. So it's capturing those areas of uncertainty quite well. And related to this domain of applicability is this concept of a global versus a local model. And global models are designed to cover as much chemical space as possible, as much chemical diversity as possible. So here's a sort of again a picture of a sort of a chemical space. You can see this hypothetical model covers most of that chemistry, and so we would expect, if it's a good model, that it would make a reasonably good prediction within all of that range of chemistry that it covers, but it has a disadvantage in that it will tend to capture sort of long-range trends across that diversity, so may not resolve between close analogs or compounds with subtle changes between them. The opposite extreme is a local model which is trained to cover a specific region of chemistry, maybe a single series or a single project or organization's chemistry. And again, if it's a good model, then we would expect it to pick up that sort of high resolution information, those more subtle differences between those closely related compounds. But of course, a local model also has a disadvantage in that, of course, as you move outside of that domain of applicability on which it's been trained, the uncertainty rapidly increases, and so it won't extrapolate well beyond that region. And so there's a sort of spectrum of models that one might consider. And typically, global models are useful if you're surveying quite a lot of chemistry and quite a diversity and trying to find sort of islands of useful and interesting properties. But of course, once you're working within your own chemistry using data specifically generated using your own protocols, ideally what you want to do is to build a local model tailored to that specific information. And I guess the ultimate question is, well, what can we realistically expect from ADME models, the sort of current state of the art? And again, I'll emphasize this is within the domain of applicability. And so typically what you would see is that if you look at the standard error in the prediction from an independent, sorry, let's just say test set using my own nomenclature, then typically you'll see for something like a log p, maybe between 0.4 and 0.5 log units are commonly reported. Solubility, again, we've seen a very nice example of that. You typically see between 0.7 and 0.8 log units. And if we're looking at, say, inhibition or affinity, PKI values, maybe up to a log unit of uncertainty. And so what does this look like in practice? Well, here's a typical solubility model built on a larger data set, in this case about, I think about 6,000 compounds, and tested on a couple of thousand. And what you can see here is the root mean square error is about 0.8 log units. Again, in line with what we just saw from Jonathan's presentation. But what does this mean in practice? Well, it means if you have a 1 and a 5 micromolar compound, this model cannot resolve between them. You shouldn't expect it to. It might have a chance between resolving a sort of a 1 and a 10 micromolar compound, but even then, it's only a probability. It's not a guarantee that it would resolve that. And just to, because we've been talking about solubility, I should point out that all this data is intrinsic thermodynamic aqueous solubility measured between 20 and 30 degrees centigrade. So not as high quality as the data set that Jonathan was just describing, of course, but really curated as best as possible from a larger collection of compounds. So there will be variability between the protocols used, and so even the correlation experimentally will not be expected to be perfect by any means. And that really comes down to the point, again, which we've heard, is that it's very important to compare like with like, because experimental measurements of ostensibly the same property with different conditions can also have a very low correlation. And again, we saw a very nice example of that, just looking at to be a simple property, but as we've learned is not, and that's solubility. Okay, so just a few minutes left. ADME models can certainly capture a lot of information, but they have significant limitations, as we've just described. So the question is, how do we actually use these predictive models to actually make reasonable decisions within the drug discovery process? And of course, this is where this big challenge of multi-parameter optimization becomes very important. Because we're not just really interested in optimizing a single property, but we know that a safe and efficacious drug that sort of makes it to the market actually has to have a balance of many different properties. And what we'd like to ideally do is move as quickly as possible from our sort of early hits or leads to this sort of sweet spot where all these properties are most likely, at least, to come together. And of course, the flip side of that is we also want to identify the situations when that balance is not possible. Essentially, when the chemistry that will give us potency against our target does not overlap with all of the other properties that we need. And while this is described as fail fast and fail cheap, so we don't want to waste time and effort pursuing this chemistry where we won't find a good drug, we want to make sure that we're doing this with confidence. Given all the uncertainty, not only in the predictions that we use, but even the experimental data, as we've seen, the last thing we want to do is to miss the opportunity to find a good drug due to all of that uncertainty in the data that we're dealing with. And so I guess the most common way that we look at this problem is often by filtering. And so just to use an analogy of finding a diamond hidden in a pile of coal, well, of course, if we were filtering maybe based on potency, maybe a high throughput screen or something like that, and we throw away the most potent, throw away the compounds that do not meet our potency criteria and take forward the ones that do. We can start to add in other properties and maybe absorption, metabolic stability. Again, we throw away the compounds that don't meet our ideal criteria. We go down through this process, not for maybe 3 properties, but maybe 10 or even 15, and we hope that out of the bottom emerges our diamond. We can put that into development and hopefully 5 or 10 years later anyway, reach that successful marketed drug. But of course it's not that easy, because often this happens and no diamond appears in the first place, and then we've got this big question on our hands. Is it because there's no diamond there in the first place, only one of these areas where these properties don't overlap? Or have we made a mismeasurement or a misprediction and incorrectly discarded that diamond? And if so, where do we go looking in this big pile of coal? And even worse is this outcome, where instead of the diamond coming out of the bottom, a lump of coal incorrectly emerges, and we spend months or even years trying to optimize it, only to have one of those dreaded, expensive, late-stage failures. And the reason I bring this up is, coming back to that question of uncertainty, how likely is this outcome? And so if we had, for example, 10 filters that were each 90% accurate in making the correct yes-no decision, although that would be a great situation to be in, the probability of that diamond emerging, even if it's there in the first place, is only 35%. So in that scenario, I'm more likely to throw away my best idea than I am to keep it. And so for that reason, this approach of filtering needs to be treated with caution, because although it seems very simple, it's a process that actually accumulates error without that being transparent. And so what we really want to do is to define the criteria we're looking for. Think about the properties that we're interested in. Again, this can be experimental as well as predicted. The criteria we're looking to achieve, and think about the trade-offs we're willing to make as well, because not all properties are equally important. Some things are critical, potency is absolutely essential, but other properties we might be willing to compromise or trade off. They don't have to be hard cutoffs. They can be so-called desirability functions with sort of soft boundaries and looking at that trade-off more subtly. But when we start to combine them, we want to consider the uncertainty in that data. So we can take our property data, experimental or predicted, assess these data against the criteria for success and their relative importance, but also take into account all of this uncertainty, whether it's predicted uncertainty, statistical uncertainty, or experimental variability, to calculate a score which will help us to rank those compounds. Essentially, well, we tend to look at this as a likelihood of success or a probability of achieving that ideal outcome. But also think about the confidence in that overall score once we combine all this underlying data. And so here's an example shown below of how we can look at that. Here we have the compounds in a data set ordered from the best on the left to the worst on the right by this overall score. And on the y-axis we have the score itself, so we can see some of these are doing very well. This is a chance of success, so about an 80% chance of success. But when we look at the error bars, when we combine all that uncertainty, we can look at the confidence in that overall score and establish when this data actually allows us to distinguish between compounds with confidence. So here, for example, these top 5 compounds, the error bars overlap almost completely. And so what that's telling us is the data we have so far do not confidently distinguish between those compounds. So if I tell someone that this is the best compound here, you'll say, well, just wait a minute. Really, we can't choose between those based on the data we have. If we do make that decision, we would need another criterion to choose between those compounds, or some better data to error bars become smaller. Whereas on the other hand, roughly the bottom 50% here, you see the error bars don't overlap, so we can reject those compounds with confidence, save that time and effort, and focus up here where the chance of success is higher. We can gather some better data or explore that chemistry further. So prioritization of compounds in terms of selection, where we want to take them forward, get some experimental data to back up those predictions, is important. But the last thing I just wanted to mention was thinking about how we can use the information in a predictive model to actually guide the next step, and how we can actually design compounds and improve their properties. And I noticed that we're really out of time, so what I'm going to do is drop out of the slides, skip a few here, and just start to think about the relationship between this mathematical function that we fit in terms of a model. This is an example of a nonlinear model. And what we do when we make a prediction is we can take our descriptor values, as I mentioned, feed those into this mathematical equation, which is represented by this surface, and the height of that surface essentially gives us the predicted value of that property. But in fact, the shape of that surface contains or provides much more information. In fact, we can think about the changes in descriptors that will have the biggest impact on increasing or changing or that property. And ultimately, in a method I don't have time to describe, we can actually map that information onto a predictive model. Because yes, we make a prediction, but of course the next question that occurs is, well, why has a model predicted a particular value? And probably more importantly, where can I actually modify that compound to have the biggest impact on changing that property? And so this idea, we call it the glowing molecule, takes that information from that very complex mathematics, but actually maps it visually onto the compound. Here you can see the red regions are predicted to have the biggest effect to increase the predicted value. The blue regions, the biggest effect to decrease it. So again, helps us to think around where can I change the structure to have the biggest impact on improving that property. Essentially getting away from these nasty black boxes where you sort of shove a structure in and it spits an answer out. So in conclusion, I think I've hopefully demonstrated it's very important to rigorously validate ADME models before using them, to understand this domain of applicability, but also once you're using the models to understand the SAR that they've captured and provide that sort of feedback. At the very least, it can actually help you to confirm that the structure-activity relationships that it's captured sort of match your knowledge and experience. And there are limitations in building good ADME models. Good quality data is very hard to come by. Again, we've heard the example, Tim asked the question about larger solubility data sets. It's very hard to come across good quality data in sufficient quantity and diversity. And also, I didn't really discuss this, but we also need better descriptors, better ways of capturing features of molecules that have a stronger correlation with these properties. I've already mentioned the current state of the art, and just to mention, you've probably seen as we've gone through, lots of references have flashed up for all the details behind this, and they're all available at the link below. So thanks a lot for your time. I'm sorry I took an extra few minutes. Mark, are you there?
Yes, I'm talking and I'm muted, which wasn't very sensible.
Wonderful.
Thank you very much, Matt. Thank you very much, Jonathan. Thank you to all the attendees. You will get an email with a recording shortly. Those of you that have seen some of the recordings before, we're actually changing them all so that they'll be on YouTube, which will make them a lot easier to visualize, because otherwise I think you've had to download them from Dropbox, so that should make life a lot easier. But thanks once again, and we will contact you shortly with information about hopefully an interesting program for next month as well. So thanks again to Jonathan and to Matt, and we'll speak to you next month. Cheers. Bye.